Thank you very much for the invitation to speak here today, and good afternoon from New York. My name is Viola Neudecker, and I'm an associate research scientist at Columbia University. Today, I would like to talk about the major findings from our two recently published studies in juvenile non-human primates after early life anesthesia exposure. I have nothing to disclose. As you all know, there's a concern and a controversy whether the exposure of young children to general anesthetics affects their neurobehavioral development. Some studies report associations between early in life anesthesia exposure and learning disabilities, deficits in general intelligence, and increased risk for attention deficit hyperactivity disorders, while other studies report none. Certainly, such controversial findings are not surprising when thinking of these studies being in majority retrospective, very heterogeneous, and containing several confounders. In contrast, there are results from 3 studies with prospective assessments that Dr. Ng combined in his recently published meta-analysis and confirmed that a single early in life exposure has no risk for deficits in IQ, but based on observations made by parents and caregivers at home or at school, exposed children have an increased risk for internalizing behavioral problems. Studies in animals also report long-term functional impairments after early in life anesthesia exposure, such as impairments in learning and memory and alterations in behavior. In juvenile non-human primates, alterations in behavior reported include increased anxiety-related behaviors. However, in contrast to the behavioral alterations reported in children, these behaviors in non-human primates have been detected by provoked testing. In addition, structural changes reported in brains of animals exposed early in life mostly relate to the acute injury, such as neuroapoptosis. In contrast, we know very little about long-lasting structural changes. So we asked 2 questions. Does early in life anesthesia exposure alter spontaneous behaviors of juvenile non-human primates in their home environment, in their social group? And are these long-term functional alterations accompanied by long-term structural changes that are evident in the brains at this age after early in life anesthesia exposure? To approach these questions, postnatal day 6 non-human primates were exposed to isoflurane for 5 hours, either 1 or 3 times on postnatal day 6, 9, and 12, while controls were exposed to oxygen only. So there were total 3 groups. After being housed with their dams in the first year, the animals were weaned and transferred into new social groups at 1 year of age. During the total 2-year life period, repeated tests of cognition and behavior have been carried out, and at 2 years of age, an age that is comparable to 6 to 8-year-old human children, so school-aged children, the animals were assessed by focal observations in their so-called home cage. To address our second question regarding accompanying long-term structural alterations, the animals were euthanized and their brains were prepared for histopathological analysis. To assess the animals' spontaneous behaviors in their social environment, we used focal observation, meaning an experienced investigator who was familiar to the animals conducted 10-minute focal observations 2 to 3 times per week for a total of 3 weeks. What was measured was the time the animals spent in one of the behaviors displayed here: close social behavior, aggressive behavior, anxiety, or appeasement behavior. The analysis of these observations revealed that the animals that were exposed to isoflurane during infancy spent less time in close social contact compared to the controls. Close social behavior was defined as behaviors like grooming or huddling or just being closer than 1 arm length to another animal. So these animals that were exposed to anesthesia during infancy were somewhat isolated and preferred to be alone. To address our second question, we decided to look for astrogliosis in the animals' brains, since brain injuries of other causes have been associated with astrogliosis, namely chronic astrocyte activation. Astrogliosis is a common response to several types of brain injuries, including trauma, ischemia, and neurodegenerative conditions like Alzheimer's disease. And astrogliosis can be identified by increased expression of glial fibrillary acidic protein, short GFAP, which is a cytoskeletal protein in astrocyte that remains chronically elevated after the brain injury. Based on previous studies that investigated brain areas indicating an acute injury, such as increased rates of apoptosis after early in life anesthesia exposure, we first analyzed GFAP densities in the primary visual cortex, also called V1. And as you can see, comparing GFAP densities, we found that 2 years after anesthesia exposure, animals showed increased GFAP densities, increased astrogliosis in V1. Based on our findings of alterations in social behaviors, we next analyzed GFAP in the amygdala, a major hub for the processing of social behaviors. And plotting the resulting GFAP densities of the three groups, we found that compared to the controls, the GFAP densities were increased 2 years after anesthesia exposure. So in summary, we found that 2 years after anesthesia exposure during infancy, the juvenile non-human primates showed decreased close social behavior when they were observed in their home environment. And at the same age, the brains of the exposed non-human primates display astrogliosis in the amygdala, a brain area that is strongly involved in the processing of social behaviors. Importantly, the functional alterations that we found in the juvenile non-human primates parallel the findings in recent clinical studies that reported behavioral alterations in the assessment of exposed children in their home or their school environment. Together with the structural alterations in the amygdala, namely the astrogliosis, our studies indicate that anesthesia exposure early in life might impair the social development and behaviors of the exposed individuals. So our future directions will focus on investigating the social development in individuals exposed to anesthesia early in life and using the non-human primate model, as this being the closest to the humans in terms of brain development and behavioral repertoire. And with this short comment on our future directions, I would like to end, and I would like to thank the Department of Anesthesiology at Columbia University and its chair, Dr. Ansgar Brambrink, who is also the head of this research, and my colleague, Jose Perez-Zogby, with whom together I did all these studies. And I also want to thank our collaborators at the National Research Primate Center in Oregon and our collaborating expert in neuropathology at OHSU. And of course, I want to thank you for your attention and your interest in our research. Thank you very much.